Several classes of hormones exist, including steroids, amino acid derivatives, and polypeptides and proteins. Steroid hormones produced by the gonads and part of the adrenal gland known as the adrenal cortex have a molecular structure similar to cholesterol. This similarity allows them to enter their target cells and interact with receptors in the cytoplasm or the cell nucleus. Once inside, the hormone receptor complexes bind to specific regions of the cell's DNA, regulating the activity of hormone-responsive genes. This mechanism of action is crucial for the regulation of various physiological processes, including metabolism, immune response, and reproductive functions. The ability of steroid hormones to directly influence gene expression makes them powerful modulators of cellular activity. Amino acid derivatives are another class of hormones, produced by the thyroid gland and the adrenal medulla. These hormones are modified versions of some of the building blocks of proteins. Like steroids, amino acid derivatives can enter the cell and interact with receptor proteins already associated with specific DNA regions. This interaction modifies the activity of the affected genes, thereby influencing various physiological processes. For example, thyroid hormones, which are amino acid derivatives, play a critical role in regulating metabolism, growth, and development. The ability of these hormones to directly interact with DNA makes them essential for maintaining homeostasis and responding to environmental changes. Polypeptide and protein hormones are chains of amino acids of various lengths, ranging from 3 to several hundred amino acids. These hormones are primarily found in the hypothalamus, pituitary gland, and pancreas. Due to their chemical structure, polypeptide and protein hormones cannot enter cells. Instead, they interact with receptors on the cell surface, initiating biochemical changes in either the cell's membrane or interior. This interaction eventually modifies the cell's activity or function. For instance, insulin, a protein hormone produced by the pancreas, regulates glucose uptake in cells, thereby controlling blood sugar levels. The inability of these hormones to enter cells necessitates a different mechanism of action compared to steroids and amino acid derivatives. To maintain the body's homeostasis and respond appropriately to environmental changes, hormone production and secretion must be tightly controlled. Many bodily functions are regulated not by a single hormone, but by several hormones that regulate each other. For example, the hypothalamus secretes releasing hormones transported via the blood to the pituitary gland. These releasing hormones induce the production and secretion of pituitary hormones, which are then transported to their target glands, such as the adrenal glands, gonads, or thyroid. The interaction of pituitary hormones with their respective target cells results in the release of hormones that ultimately influence the organs targeted by the hormone cascade. Constant feedback from the target glands to the hypothalamus and pituitary ensures that the activity of the hormone system remains within appropriate boundaries. Negative feedback mechanisms are common in hormone regulation. Hormones released by target glands affect the pituitary gland and/or hypothalamus, turning off the hormone release cascade when certain blood levels are reached. For example, the progressive reduction in sensitivity of the hypothalamus and pituitary to negative feedback by gonadal steroid hormones plays a crucial role in sexual maturation. Although negative feedback is more common, some hormone systems are controlled by positive feedback mechanisms. During a woman's menstrual period, increasing estrogen levels temporarily stimulate hormone release from the pituitary and hypothalamus, leading to ovulation. This positive feedback loop requires a specific threshold level to be turned off, maintaining a stable system.